Good evening. I'd like to start with a question. Do you know what have in common cultivated meat, COVID-19 vaccines, cancer treatments, fermentation processes, the face cream I'm wearing, the yogurt I had this morning, or a biogas plant? All of them are produced in bioreactors, which are the basic, or fermenters, which are the basic equipment to manufacture food biotech, to manufacture many biological compounds. This is a very large market because it applies to many different sectors, but it presents quite a few problems. So for example, they are expensive. Processing times sometimes are long because they are living microorganisms. So we need to provide them with oxygen, we need to provide them with nutrients. And sometimes the oxygen transfer is not good enough or is not high enough, and then we can get long processing times. Yeah. Other times, clients came to us with the problem of quality, because sometimes the culture inside the bioreactor was not homogeneous, or maybe if the agitation was too strong, maybe the cells got damaged, so they got shear problems. Energy consumption can also be a problem when working at the industrial scale, because bioreactors can be very large and energy demand can be high. Also, in the scaling processes, sometimes there are too many steps and that takes time. At the end, this becomes a problem of mixing and aeration. Which is our solution? Well, based on our knowledge on fluid mechanics, on aerodynamics, on complex mathematical models, we have designed a software simulation, numerical simulations, with which we can know what is happening inside the bioreactor. So we can see the oxygen transfer at each point, shear forces at each point, and how everything behaves inside. And with that, we have designed an agitation and aeration system, which we have patented. That's the standard one, with which we can retrofit our clients' bioreactor to improve the performance of those bioreactors. We can also offer the whole bioreactor with our system inside. Which are the advantages? With the numerical simulations, we can reduce the process of scaling, because we know how everything behaves in a smaller bioreactor, and so we can predict how it is going to behave in a larger one. We can also reduce processing times thanks to a better mixing and thanks to a higher oxygen transfer. We have reduced it up to 50% for some of our clients. We also get a higher quality thanks to the better mixing, thanks to a lower shear, and we can also reduce energy consumption inside large bioreactors. So at the end of the day, we can reduce time and money for our clients. This is an example. The video is not working, but on the left is the bioreactor of one of our clients. He came to us because he had a problem of oxygen transfer. It was a first brand bioreactor. The bubbles were too thick and they were not homogeneously distributed inside the bioreactor. So after we retrofitted our client's bioreactor, we got a much more homogeneous gas inside the bioreactor. The bubbles were thinner, but not too thin, because if they are too thin, they can damage the cells. So we calculated it so they were the right size for that culture. So we got an increase of oxygen transfer of 90% and a reduction of the processing time by 50% in this case, and with less shear on the culture. So these are other stories that we got for some other clients. We can work with different sizes of bioreactors. We have worked with bioreactors of less than 1 liter to huge bioreactors like 200,000 liters for a petrol company that was producing bioplastics. This is some internal validation. This is a comparison we made with the very, very first brand of bioreactors. Probably we have it in mind, the name of that brand. And we compare their system with their bioreactor with our system inside. And you can see that we can increase KLA, which is a measure of oxygen transfer, by up to 82%. Yes, so we can increase oxygen transfer quite a lot, which is very good in the case of fermentation processes or for yeast or for cultures that demand a high oxygen transfer. On the right-hand side of the table down there, the improvement is lower, but that is really very infrequently to use that, to have that situation, because you would have lots of foam, so that is not much use. We can also decrease shear. We have a different model of agitation system that is more aerodynamic and it cares of the culture, right? So we can decrease the shear on the culture. Like in this case, we could decrease the shear and at the same time, we increase the KLA. So we improve both parameters, and at the same time, the client could lower the speed of the bioreactor, so the shear was even lower than that. Who are our clients? Well, they are bioreactor users and also bioreactor manufacturers who can include our systems in their bioreactors. What are we looking for? Well, we have some collaborations with, or we are dealing with some European and USA bioreactor manufacturers. We have also some clients, bioreactor users, and now we are looking to the Asian market, in particular to the Japanese market, because there is a very, very large market here, and we would like to make collaborations with bioreactor manufacturers and also with bioreactor users. Thank you very much. This, I finished my presentation. Thank you.
Thank you.
I will be at the booth there in case somebody wants any questions or want to talk to us. Okay, thank you very much.
Great, thank you. So the next speaker is Floris Engelhardt, co-founder and CEO of Kanos Therapeutics, DNA nanotechnology and synthetic biology for gene delivery.
Oh, this is my, okay. Thank you everyone for being here today. I'm standing here in front of you today because I am a proof that you can become a CEO when you were just a researcher. Kanos Therapeutics is a company that we started 2.5 years ago, and it's actually a gene therapy and cell therapy company. But I must disclose right at the start, originally my PhD and my research work was not in gene editing. So I come to this field with a background in DNA nanotechnology and biophysics. And with this background, I stumbled over an issue in gene editing that is quite annoying. And that is that if you think about it, and all what currently cell therapies and gene therapy companies do is that it's easier to block gene function than it is actually to repair gene function. But if we think about genetic diseases, only 30% can be solved with small edits, whereas the other 70% need larger insertions, or you would need to think about replacing whole long genes. It's kind of if you wanted to repair or edit a book and you're like, oh, we can only edit one letter at a time. Maybe you should be able to copy and replace whole sentences in order to edit a book. And so from a DNA perspective, we then looked at the market. And we saw that there's a lot of innovation obviously going on, but if you think about DNA insertions on the long run, you can see that virus systems are normally focused on random insertions, and they have come across very much adverse effects. And then if you think about nucleases or next gene editing, which would be non-viral options, there's huge problems there with low insertion rates and also limitations of insertion efficiency. So this billion-dollar market is basically constrained across 3 different big topics: safety, efficacy, and scalability. At Cano, we looked at where is innovation currently happening and where do we have to go. And there's a lot of innovation when it comes to delivery, novel delivery tools, and editing tools. Where there's no innovation is the DNA repair template. A lot of people currently look into the RNA field, but most people working with DNA still work with double-stranded DNA plasmids because they're cheaper and better available. But we know that DNA toxicity is no easy thing to overcome. And as long as we work with double-stranded DNA, the immune system will react to it. So what we did is we said, well, for a repair template, we actually don't need both copies. We don't need double-stranded DNA. We could do the same thing just using the single-stranded DNA molecule, which is just one copy. So we can engineer genes into a circular single-stranded DNA form that is safer and more flexible, and where we can really think about how can we engineer the cargo adapted to the delivery. And so when we did that, we actually, and I hope this is kind of able to see, we compared our single-stranded DNA against what we would normally, like the standard would be used as a double-stranded DNA. And we already, just by transfecting the single-stranded DNA and then the double-stranded DNA in same amounts, could show that our single-stranded DNA keeps the cells happy and healthy, whereas double-stranded DNA toxicity causes the cells to die. And so this was the first step for us to really understand that single-stranded DNA is the safer material, and we just need to learn how to engineer it for use in genetic therapies. So we started working with a collaborator, and we wanted to look into non-viral cell therapies, because a huge problem for cell therapies, especially cancer immunotherapies, is toxicity because of viral vectors, and people want to go to non-viral approaches. So this experiment was, we use a single-stranded DNA template with a therapeutic CAR gene, and we use CRISPR to insert it into primary T cells, because that's what the patients would need. And we could compare that again against standard DNA templates. In this experiment, we could show that our circular single-stranded DNA actually performs 4 to 8 times better than a linear single-stranded DNA that is not as stable, or a double-stranded DNA molecule, which would be the industry standard. And so this is a huge difference that could be made for patients, because if you think about a cancer patient that needs an immunotherapy, they're already at the latest stage. They don't have many immune cells to collect, engineer, and give back to the patient. And even if you wanted to do allogeneic therapies, the amount of times you have to recycle the cells, like 4 to 8 times more cells right from the start, is a game changer. It makes non-viral therapies possible. And so we really believe that by improving the cargo, we can enable across all different gene editing therapies to make insertions possible. So we're building now end-to-end capabilities to fit the 3 different problems. Number 1, safety. We're looking into 3-dimensional design analysis, because the huge opportunity we have here is that if anyone in the room worked with RNA, we know RNA makes 3-dimensional structures. DNA, we just know it, the normal helix, but we have single-stranded DNA. So we can think about 3-dimensional structures, design, and functionality. We already have shown that it is much more efficient as gene insertion molecule. And now comes the big difference for us and for me as my background. The reason I started this company is because I found out that the one point why no one has already used single-stranded DNA, especially in kilo-base length, is that there is a huge manufacturing bottleneck. You cannot buy that easily. So what we found is that we can use synthetic biology and bacterial fermentation in order to use bacteria to make customized single-stranded DNAs that we design. And it's scalable and it's pure. And so we now have unlimited access to these molecules that no one currently has access to on a cost-efficient basis. And that allows us to start studying it, working with it, improving it. This led us to the point that we have a lot of company market pull. We had companies reaching out to us, really interested in using it across different modalities. We have in vivo gene therapies, CAR T cell therapies. We're not working on vaccines right now. I know people have reached out to that, but we're focused on the cell and gene therapy space. But it's really understanding from our point of view is like, how can we enable the most clinical programs using our single-stranded DNA molecule? So we're working on internal programs in CAR T and HSPC environments, and we're currently looking for expanding our collaborations. So why am I here today? Just because of that reason. As you might have heard, I'm not US American. I actually am German. I came to the US in 2020, just for my postdoc. And so it was fairly easy to make the connection to Europe, to European companies, to American companies, and MIT supports you a lot. But I sadly have to admit that we don't have any partners in the Asian realm. So the reason I'm here today is because we have a scalable manufacturing process for kilo-based long single-stranded DNA. We're currently working on process development, GXP, and quality systems, but we don't have clean rooms. So we're looking for cGMP manufacturers that are interested. And on top of that, we're looking for non-viral delivery companies that are interested working with us because they want to switch from double-stranded DNA to single-stranded DNA, or from viral delivery to non-viral delivery, and want someone who understands the cargo to really design the single-stranded DNA to have higher insertions rates. I truly believe that only through partnerships we can complete that mission, because ultimately we all have DNA, and even though we're all perfect, our DNA often is not, and these can lead to fatal errors that just are DNA errors that can hit anyone across the world. So thank you for listening to me, and I'm happy to talk later at the booth.
Thank you, Floris. The next stop is on AI-enhanced imaging and large language tools for drug discovery by Kevin Christopher, CEO of QuantiScope. Thank you. Thank you, Kevin.
Okay, thank you very much. An absolute pleasure to be here. My name is Kevin Christopher, and I'm one of the co-founders of QuantiScope. We launched out of my co-founder Juan Caicedo's lab at the Broad Institute, based around technology developed there in support of some of the companies in this room through industry consortia. And so I thank you as well. Hats off to those companies. We are automating image-based intelligence technology for drug discovery and development. Now, what does that mean? Basically, if I take out my phone and unlock it by looking at it, it is taking my phenotypic profile. It says, okay, here's a man, blonde hair, few facial scars from being an adventurous boy, a nose that's been broken a couple of times. That's Kevin. And so I'm going to provide access, similar to how we pay for things with our face now as well. A little bit of an extension of that technology comes from measuring my phenotype against a standard to determine, am I sick or am I stressed out right now? Which I'm not right now because you're all being so lovely and welcoming to my talk. So not a problem right now. But we're essentially taking those fundamental concepts and we're applying them to images of biological experiments. And so our core technology was developed based upon high content image analysis. So high throughput workflows, drug screening. And what our technology does when it's presented with images in these workflows is it essentially uses a three-pronged process of segmentation, feature extraction, and batch correction. So the first step is identifying in an image what's noise and what do we care about? What are the cells? And more importantly, where are the nuclei within those cells? Because that gives us sort of our geolocation of where all the other points of interest would be. Our platform then uses feature extraction, and an AI component of this is essentially determining which features it wants to extract and measure. And so sometimes it might be a couple of hundred morphological features of the cell. Sometimes it might be over 1,000 to measure against one another in using those morphological features to create drug signatures. So essentially morphological signatures in response to the drug or the cellular perturbation. And so within one image, we may have hundreds of cells, and each of those cells receives its own phenotypic signature. And then we can use that to create hierarchies and ranking. And by quantifying some of that information, we can actually unlock intelligence within the biological systems themselves. And so one example from earlier this year, I mentioned earlier, we're automating this. That's really important because at the Broad Institute, various researchers involved with this type of technology have released some of the data sets and some of the models for the industry and academic world to uptake, but it's been slow to develop. Even companies with vast resources, with whole imaging teams within the companies, struggle with how to incorporate this into their workflows, much less smaller teams in academia or small biotechs or in developing parts of the world. And so this is a pilot we ran with a multinational pharma earlier this year, and they presented to us about 240,000 images featuring about 7 million cells. And they had already kind of run their analysis of this library over many months, and they had tried imaging as well. But one of the capabilities they did not have was analyzing bright field images. So their capabilities stopped at analyzing cell painting assays, essentially stained images. And so the challenge to us was, can you get the same ranking through just image analysis? And furthermore, can you analyze bright field images and come up with the same data? And without any knowledge of the experiments, any metadata whatsoever, just the images, we were able to correctly pull out the lead candidates out of this library in just a few hours of computational time. And that's how powerful this type of technology can be in a workflow. It can drastically reduce times, de-risk the process, and provide intelligence that other methods simply can't. And so this was just an exciting development for us. Some of the other applications that are possible include, for instance, measuring the phenotypes in lipid nanoparticle processes in manufacture and in quality control. And so that same type of approach can be taken to create a simple image-based methodology for measuring changes in the lipid nanoparticle nucleic acid conjugates along a supply chain. And this is really important because we've seen some data recently about the mRNA COVID vaccines and specifically tying adverse effects to batch inconsistencies around the world. And so this is a critical need, easy ways to use imaging to conduct these analyses along the supply chain. Another area where this would be exciting for us to participate with supporters like you and partners like you would be in cell line selection. The biologics sector of the industry is going to account for maybe nearly 40% of the pharmaceutical industry by 2030. And one of the tedious workflow processes is selecting an optimal cell line for production. And so We've seen partners who go through thousands and thousands of potential clones trying to sort through. We can use phenotypic analysis and automate this process as well. So these are the types of opportunities that I see in making this trip. It's not just for the whiskey, although the whiskey is very, very good, but it's the opportunity to speak with you, to meet with you, to talk about our vision and our capabilities, and to help us expand our platforms. This is a screenshot of the platform that we've developed for high content image analysis in drug screening. So we are also looking for beta testers there as well. We want as much feedback as possible over the next 6 months before a product launch. So definitely invite any conversations around that as well. Thank you very much.
Thank you, Kevin. 続きまして、I'm introducing Carl, founder and CEO of Chiba Bio, on new delivery modalities for RNA therapeutics. Carl, よろしくお願いします。
皆さんありがとうございました。そう、多分皆さんはよく覚えてます。COVID-19 のワクチンもらったときに、大変腕が痛くて、1、2間の熱があったかもしれない。私もちろんもらった。その副作用は海管からじゃなくて、RNA からじゃなくて、それはlipid nanoparticle の影響だった。多分、パンデミックの時代、そのような副作用は大丈夫。けど、普通のワクチンか、次の時代の RNA 薬、例えば、intravenous injection は、そのような延長は大変かもしれない。Chiba Biotech は、その問題を探す、ソリューションを探します。Okay, that's about the edge of my Japanese. So, I'll try to do a nice, kind job to our translators and introduce Chiba Biotech. So we started in 2018 after 5 years of research within the same lab that gave birth to Moderna. And since that time, we were able to attract a number of US grants and also international grants. And basically today, after the pandemic, we're pretty much revenue positive just from grants, which is a unique characteristic of biotechs today. And more recently, just as early as last week, we issued a press release mentioning that we received a grant from BARDA to do our next generation of therapeutics. So we started our program in vaccines over the pandemic period, and now we're advancing into therapeutics. So what's the problem? I had mentioned in Japanese, I'm sorry, maybe most of you didn't understand. The problem with today's delivery of RNA is primarily the inflammatory response. But in addition to that, certainly for vaccines, it's the stability. If you may recall, we had to keep those RNA vaccines at -80 during transportation. Maybe something we can do here in Japan or the United States, but third world countries, it's a challenge. Also, a lot of the lipid nanoparticles, they dissolve to the liver, which for some applications may be okay, but later on I'll talk about some new next generation applications. And then it's a very complicated patent space. Every company's suing each other. So what Chiba has designed out of MIT is a novel dendrimer nanoparticle that is more stable, lower inflammation, avoids the liver, and it's a new area of IP. So let me explain this a little bit more. We've developed our nanoparticles to deliver and disappear. They're comprised of a dendrimer scaffold with a large number of positive charges that can adhere to the negative RNA backbone. And along with that, we've been shown that our nanoparticles can be designed to attract to a particular organ of interest. This is an early indication or an early example of our vaccine program, in which two example RNA nanoparticles, we call them RNA-able, had greater gene expression, but at 24 hours lower inflammation. Oops, multitasking. I'll jump to the next slide. We also show that we can focus on particular organs of interest. So your typical lipid nanoparticle, this one is the SM102, which is the one that comes from Moderna, well known to target the liver. Ours, we have about 250 delivery molecules in our library. Each one can be designed for certain purposes. Recently, we've been working on heart delivery. Very exciting for us to be able to get a RNA payload to the heart, and this is through intravenous injection, avoiding the liver. Lungs, this is what our BARDA contract, which we announced last week, focuses on. We can also, through intravenous injection, deliver RNA that goes to the lung. The case point here would be if you are immune compromised or having other medical problems, for you to get a shot of an RNA application through your nose could be very challenging. This is an application that will go intravenous while you're in the hospital, a particular interest of BARDA. And then the spleen. We have a number of other targets, but spleen we're looking at for immuno-oncology applications. This is our pipeline today, mostly focused at the initial stages on vaccines, as you see here. Again, we came through the pandemic. Our most advanced is a NIH SBIR, which is a Small Business Innovation Research Grant, is now a phase 2, meaning a phase 2 grant, not phase 2 regulatory. That's our most advanced program. We're also working on Japanese encephalitis vaccine virus that's sponsored by CEPI. CEPI is a multinational program called the Coalition for Epidemic Preparedness and Innovation. And then we also have a unique twist to our work. We're also doing emergency use animal vaccines. So this is a little bit of the One Health zoonotic vaccine program that we're being funded with, and that's very exciting for us. Now we're looking at therapeutics, as I mentioned. The siRNA is sponsored by BARDA and open for discussions, and maybe afterwards in the back room you can have a conversation with us. We're also looking for partnerships on our therapeutics. This is our team of international specialists with experience in manufacturing, virology, and also lipid nanoparticle and dendrimer nanoparticle applications. And I'm here today to practice my Japanese. Now, we have a colleague in a small operation down in Australia where we're focusing on animal vaccines, but we're looking to expand to therapeutics, expand to other geographical regions, and hopefully to Japan. So I look forward to talking to you. I'll be out back. Thank you.